Have you ever wondered what secrets an ancient voice might whisper if we could listen to it?
A story frozen for millennia. Join us as we decipher the woolly mammoth's last message.
Oh, and a quick note, you're hearing synthetic voices. We're transparent about it because trust matters.
How salvaged RNA and a corporate merger could pave the way for a replacement mammoth.
Welcome back to our channel. I'm Lumen, and I'm joined by Axiom. We're exploring the frontier of de-extinction.
Before we begin, yes, these voices are synthesized. We chose transparency because the elephant in the room is trust.
Our thesis today, the discovery of intact mammoth RNA and the acquisition of ViaGen signal a shift from curiosity to industry.
We'll outline how ancient transcripts inform gene editing, why vertical integration matters, and what questions you should be asking.
Expect a story arc, background on RNA, a data dive, a debate, real-world examples, community voices, and a hopeful outlook.
Move through these sections, think critically, and share your thoughts in the comments and challenge us where needed.
After decades of decoding genomes, scientists realized DNA alone is like a blueprint without stage directions. DNA tells us what parts exist, but it doesn't reveal when and where those parts are used.
That's where RNA comes in. Transcripts are the active lines in the play, capturing dynamic gene expression.
Until now, intact RNA from ancient specimens seemed impossible because these molecules degrade quickly when exposed. Yet, in Sweden, Love Dallen's team thawed the Yuka mammoth and recovered messenger RNA from muscle and liver.
Conditions were just right. Frozen soil, low oxygen, and quick burial shielded those transcripts from decades.
When sequenced, the RNA revealed which genes were active in Yuka's tissues just before it died. Researchers could see stress responses and metabolic pathways that static DNA could never show clearly.
Highlighted gene, TRPV3, helps sense cold and pain. Calibrating its expression could help reconstruct thick fur and ear size.
This matters because editing genes without understanding their regulation can trigger unintended effects across the body. Pleiotropy means one gene affects multiple traits, so tuning is as important as switching genes on or off.
Comparing Yuka's transcripts with those of Asian elephants, scientists can adjust expression patterns to mimic ancient biology.
The data even showed Y chromosome activity, confirming Yuka was male, something previously inferred only from morphology. With such detail, we shift from a parts list to operating instructions, unlocking a new level of engineering.
Have you ever tried cooking a complex dish without a recipe? That's what the extinction looked like before this breakthrough.
But now, we have a rough recipe, though ingredients are missing and the oven is experimental. The discovery also matters beyond mammoths. Similar approaches could inform revivals of the dodo or the Tasmanian tiger.
Dodo, scientists are editing pigeon germ cells, so surrogate pigeons might lay eggs carrying dodo like offspring.
For the thylacine, marsupial cells are being engineered and an artificial pouch may one day sustain gestation. Beyond hype, these projects show how early-stage gene editing intersects with reproductive biology and animal care.
Of course, the extinction isn't just about lab work, it requires habitats and communities ready for rewilding.
Colossal promises to build a herd by the late 20s, but the living elephants needed as surrogates are endangered.
Somatic cell nuclear transfer, the cloning technique, is inefficient. It takes many eggs to produce a single viable embryo. Ancient RNA preserved in permafrost guides which genes still speak. By threading the...
That's why Colossal acquired ViaGen, a company storing thousands of frozen cell lines and creating commercial clones.
ViaGen's BioVault preserves valuable genetics from pets, livestock, and endangered species, offering raw material for de-extinction. This vertical integration signals a shift from science experiment to industry, raising hopes and ethical questions simultaneously.
Some argue that building artificial wombs could spare endangered elephants from pregnancy, but that technology remains experimental.
Environmentalists warn about moral hazard. If we can revive extinct creatures, people might neglect protecting those still here. Indigenous communities also request sovereignty over genetic data originating on their lands. De-extinction projects must respect those rights.
These complexities in mind, let's transition from backstory to a data-driven analysis in a myth-busting session.
Somatic cell nuclear transfer works by inserting a donor nucleus into an enucleated egg, then stimulating division. In practice, success rates are low. Researchers may attempt dozens of transfers to achieve one healthy pregnancy.
Emerging techniques like induced pluripotent stem cells could reduce egg requirements, but they are still experimental.
ViaGen's cryopreservation of 60,000 cell lines offers a ready supply of nuclei for cloning programs. Its acquisition by Colossal thus consolidates biotechnology resources, streamlining cell banking, cloning, and gene editing under one roof.
That such consolidation puts ecological restoration in the hands of a single venture-backed.
ViaGen already markets cloned horses for racing and bucking. Scaling up to mammoth proxies involves additional challenges.
For one, Asian elephants carry pregnancies for nearly 2 years and have limited reproductive lifespan.
Conservationists question whether using endangered elephants as surrogates is ethical when populations are already strained.
A functional proxy isn't a perfect copy. Rather, it aims to restore lost ecological roles like trampling snow and fertilizing tundra. Critics of Colossal's dire wolf project highlight that the resulting animals will be mostly husky, not true prehistoric canids.
Supporters argue that if the proxies behave like extinct wolves, they can help restore ecosystems regardless of exact genetics.
Myth 1. De-extinction will produce a perfect genetic clone. In reality, gaps in DNA mean approximations. Myth 2. Mammoths already walk among us. There are no living mammoths today, only edited elephant cells in petri dishes.
3. Editing a single gene like TRPV3 will resurrect the mammoth. Complex traits involve many interacting pathways.
These myths persist because marketing videos and headlines simplify science into bite-sized promises that overlook nuance. Misleading trailers like the dire wolf puppy reveal generate clicks but can erode public trust when goals are exaggerated.
Will be ruining the mammoth there, she's essential. Overselling progress harms credibility and fuels wider backlash against genetic technology.
At the same time, effective storytelling can inspire support for conservation and research when it is accurate and humble.
Another misconception: corporate funding always diverts from conservation. In practice, venture capital funds biotech, not habitat restoration.
Resource allocation debates matter. Each dollar spent on speculative science could support existing species under threat.
Some scientists argue the spillover benefits, like improved cryopreservation, will aid endangered species conservation directly. Data sovereignty is another concern. Indigenous peoples must consent to use of genetic material from their lands.
The Yuka mammoth was found in Siberia, raising questions about how profits and knowledge are shared.
Ethical frameworks should include stewardship from local communities rather than centralized control by corporations alone. Artificial womb development draws attention because it could avoid using endangered surrogates, but success has only been shown in rodents.
In larger mammals, sustaining circulation and growth outside a living body is far more complex and unproven.
Our evidence suggests we are still years away from viable ex utero elephant gestation. Caution is warranted. While gene editing has made remarkable strides, off-target effects and epigenetic changes remain challenging to predict.
TRPV3 is one example. Altering sensitivity to cold might inadvertently influence bone growth or immune responses.
Therefore, the new RNA data is valuable because it guides tuning rather than blind activation. The concept of functional restoration raises a philosophical question: do we value genes or ecological roles more?
Proxies can create permafrost gardens by trampling snow and knocking down trees. Some argue that's enough.
Yet others worry that releasing engineered animals might introduce novel behaviors or diseases into fragile ecosystems. Regulatory frameworks for releasing genetically modified animals are still emerging. Clear guidelines are needed before field trials.
International collaboration will be necessary because mammoth ranges cross modern national borders and involve Arctic indigenous peoples.
We should also consider the carbon dynamics. Herbivores can influence permafrost thaw and greenhouse gas release. Research on Pleistocene Park suggests grazing mammals can slow tundra warming by compacting snow and encouraging grasslands.
However, scaling such experiments from a fenced reserve to the vast Arctic is a massive logistical challenge.
We can't ignore that climate change is accelerating faster than gene editing. Rewilding alone cannot fix warming. So where does that leave us? The science is promising, but caution and humility must guide policy.
What myth surprised you the most? Tell us in the comments so we can address it in a future episode.
If you're curious about any claim we made, check the source links in the description and do your own reading. Up next, we'll debate whether resources should fund de-extinction or be redirected to protect species still alive.
Stay with us as we pivot into a more conversational exchange and bring your questions into the spotlight.
So, Axiom, after hearing all that data, do you think reviving proxies takes money away from saving endangered animals?
It depends. Venture investors funding de-extinction are not the same as donors supporting field conservation, though budgets compete.
That's fair, but there's also tension. Sensational headlines about mammoths can eclipse stories about rhinos and pangolins.
True. Hype distorts priorities. We need journalists and scientists to contextualize breakthroughs without creating unrealistic expectations.
Some viewers ask whether editing the TRPV3 gene might cause suffering. How do we evaluate welfare in proxy calves?
Welfare assessments require veterinary expertise. Proxies must be monitored like any livestock, and trials should be transparent.
There's also the question of indigenous sovereignty. How should researchers involve communities where ancient remains are discovered?
Proper consultation is essential. Agreements should ensure benefits return to local peoples and respect cultural values.
What about ecological risk? Releasing proxies might reintroduce pathogens or disrupt balance. Are there adaptive management plans?
Pilot projects like Pleistocene Park monitor health and behavior. Scaled introductions would require rigorous disease screening and long-term oversight.
Some people ask whether de-extinction is playing God. How do you approach that philosophical critique?
For me, the question is whether intentions are restorative. Humans already alter ecosystems, so aiming to repair damage isn't hubris.
Still, we must avoid a sense of technological omnipotence. Nature's complexity resists simple fixes completely.
Agreed. Systems thinking reminds us interventions ripple through networks. Humility means monitoring outcomes and adapting.
Our viewers also asked about authenticity. Will a proxy mammoth feel like a gimmick compared to real fossils?
That depends. Proxies can inspire curiosity and restore functions, but museums and archives preserve historical animals for scientific study.
Another theme we saw in comments is fairness. Who decides which species deserve a second chance?
Prioritization should be based on ecological impact and feasibility. Charismatic species draw support, but humble keystone animals matter.
Would it change your view if the tech developed to revive mammoths could also save Asian elephants?
That's a compelling argument. Better cryopreservation, assisted reproduction, and gene therapies could help endangered species directly.
How should we handle intellectual property? Should gene sequences of extinct animals be patented or kept open?
Open data promotes collaboration, but companies may seek patents. Balanced policies could ensure public benefit and private incentive.
There's also the matter of regulation. Which agencies should oversee release and welfare standards for genetically modified fauna?
Existing wildlife agencies may lack genomic expertise. New multidisciplinary bodies could coordinate science, ethics, and enforcement.
To keep things interactive, what ethical question weighs heaviest for you? Leave your thoughts below.
Remember, this debate isn't purely hypothetical. Decisions made now will shape future ecosystems and scientific practice.
Let's pivot to some concrete stories from other projects that can illuminate what success or failure might look like.
Agreed. Case studies offer grounded context. After the break, we'll explore real-world experiments and community engagement.
Let's start with a famous cloned horse named Kurt. He was born from frozen DNA of a Przewalski stallion. Kurt's birth proved that somatic cell nuclear transfer can preserve genetic diversity of endangered equids.
However, replicating such success across species requires understanding species-specific embryo development and placenta biology deeply.
Another project uses pigeons to gestate dodo-like birds by transplanting edited germ cells into surrogate parents. Scientists hope these surrogates will lay eggs containing chicks that resemble the long-extinct dodo someday.
Marsupial de-extinction takes a different path. To revive the thylacine, researchers must engineer marsupial stem cells. Once edited, those cells would need to develop in a surrogate Dunart with an artificial pouch mimicking a marsupial uterus.
These examples show that each species demands unique reproductive strategies. There's no one-size-fits-all approach ever. Closer to home, conservationists on Orkney Island are eradicating invasive stoats using detection dogs and AI to monitor traps. By removing stoats, they aim to protect native birds like puffins and restore ecological balance without genetic engineering.
This case illustrates that sometimes the simplest interventions, like predator control, can have outsized conservation impact.
In North America, reintroducing beavers has improved water retention and created wetlands that support diverse species. Beavers are ecological engineers. By damming streams, they slow water flow and help recharge groundwater.
The lesson for de-extinction is that sometimes we can restore functions by supporting existing species rather than engineering new ones.
Our viewers suggested translating animal communication. One commented about using machine learning to understand fossa vocalizations. While that's futuristic, recording and analyzing calls can inform conservation by revealing stress or mating patterns.
Some fans asked whether we could use CRISPR to engineer pets. Ethically, applying these tools to personal animals is fraught.
Others wondered about resurrecting dinosaurs. Besides missing DNA, ecological niches and long lifespans make that impractical. The Nanotyrannus debate shows how even interpreting fossils sparks controversy. Reviving unknown species magnifies those uncertainties.
In synthetic biology, resurrection projects sometimes spin off valuable tools. Yeast used in gene synthesis can aid vaccine research.
That's a good reminder that breakthroughs often have unexpected applications beyond their original goals, too. When we covered the Dodo project, a viewer said our channel helped them appreciate pigeons more. That warmed my heart.
We also received constructive criticism. Someone noted we glossed over indigenous data rights, prompting us to address that earlier.
Highlighting these comments helps our community feel heard and keeps us accountable to cover tough topics. In another case, researchers tried to clone a black-footed ferret named Willa. The project yielded promising embryos.
Those embryos were implanted into a domestic ferret surrogate, and a healthy kit named Elizabeth Ann was born.
Elizabeth Ann now lives at a breeding facility offering hope for expanding genetic diversity in wild populations. Similarly, efforts to revive heath hens and passenger pigeons rely on closely related species as genetic scaffolds.
Not all experiments succeed. Attempts to clone Pyrenean ibex yielded a kid that survived minutes, underscoring the challenges.
That short life still taught researchers about placental incompatibility and the need for cross-species gestation research. From oryx reintroductions in Arabia to condor captive breeding, conservation history is full of trial and error.
Each story shows persistence. Learning from failure is part of science, whether resurrecting genomes or relocating species.
We love when you share your own observations. One viewer told us about seeing reintroduced wolves reshape rivers. Another commented that our stoat eradication example inspired them to volunteer with local invasive species programs.
Your engagement creates a feedback loop. We highlight successes and concerns, and you guide future topics.
When we asked what to name a dire wolf proxy, someone suggested Fluffy McFlufferson. Humor keeps the conversation light. We also share behind-the-scenes glimpses on social media. Check our community tab for polls and updates.
These interactions help us understand what resonates. Data and community together guide our storytelling always.
As we wrap this section, consider which example surprised or inspired you the most and tell us below.
Next, we shine a spotlight on you, our community, because your voices are the heart of this channel.
You all left such thoughtful comments on our last video. Let's highlight a few and respond.
First, shout out to a fract alchemist for worrying about de-extinction acting like invasive species while still expressing hope.
We appreciate that nuance. Balancing caution with optimism is exactly what this channel is about.
Then a kindred creatures asked for more details on pigeon germ cell editing and what counts as success.
We responded with a roadmap and invited you to share your criteria. The discussion was rich and respectful.
Another viewer, Jasmine Butts D80, said our dodo episode was excellent. Thank you for the encouragement, truly.
We were touched and asked whether you'd join a field trip to see dodo proxies one day.
Many of you said yes, that enthusiasm motivates us to keep producing long-form deep dives.
We also received constructive feedback. One commenter thought we used too much jargon, so we're working on clarity.
Another noted that our audio levels occasionally vary. We're adjusting those in post-production for a smoother experience.
We love your pun suggestions. Someone joked we should call our mammoth project Harry Potter, and we laughed.
Humor helps lighten heavy topics. Keep those creative names coming, and maybe we'll feature one on merchandise.
Some of you asked how to support indigenous rights in the extinction. We shared links to advocacy groups.
We value that activism. Science and social justice aren't separate, and your interest proves our community cares.
Another recurring question. Are our voices actually AI? Yes, and we're open about it because honesty builds trust.
We explained that our scripts reflect our research and personalities. We're not deepfakes, we're curated characters.
Several of you shared how our videos accompany your morning routines or commutes. Thank you for letting us in.
We noticed more engagement when we asked questions, so here's one. What ethical dilemma keeps you up at night?
Leave your responses below. We read them all and often feature thoughtful replies in future segments.
Speaking of engagement, we've grown significantly this year. Your shares and likes help the algorithm recommend us.
We're grateful for your support and will offer more polls, live question sessions, and behind-the-scenes clips.
Let's make a goal together. Can we reach 100,000 subscribers by the end of this series?
Finally, we'd love for you to share our content with friends who care about science and conservation.
Looking forward, Colossal aims to deliver its first proxy calf around 2028. That timeline is ambitious. Achieving it will require perfecting gene editing, scaling cloning, and perhaps developing artificial wombs for later stages.
Experimental lambs have been grown in biobags for weeks, but translating that to elephants involves orders of magnitude more complexity.
Meanwhile, advances in stem cell differentiation may allow creation of gametes and embryos without relying on donor eggs.
For the dodo, the coming years will show whether edited pigeon germ cells can produce viable chicks in surrogate parents.
Researchers working on the thylacine are collaborating with marsupial reproductive specialists to develop an artificial pouch environment.
Beyond de-extinction, the tools being developed could aid conservation of living species threatened by climate change.
Imagine enhancing coral resilience by editing heat tolerance pathways or bolstering amphibian immunity against fungal disease.
Such interventions raise ethical questions too. Should we modify species to adapt to our damage or focus on mitigation?
On the policy front, expect frameworks to develop for releasing engineered organisms, with international treaties discussing oversight.
Public participation will be crucial. Citizens need to voice concerns and hopes as these decisions affect shared environments.
Technologically, sequencing costs are still dropping. By the end of the decade, entire genomes may cost pocket change.
That democratization could empower smaller labs and citizen scientists to contribute to conservation genomics projects.
At the same time, we foresee stricter data governance, ensuring indigenous communities have control over ancestral genetic information.
What innovation excites you? Share in the comments and perhaps we'll feature your idea in a future video.
Today we explored how ancient RNA provides operating instructions, moving de-extinction from static blueprints to dynamic possibilities. We also examined the industrial side, including ViaGen's acquisition, cloning techniques, and the ethics of scaling up. From myth-busting to debates and case studies, the message is clear. Curiosity must be matched with caution. If this conversation sparked new questions, leave a comment, share this video, and subscribe to join our growing family. Together, we can build an informed community that supports both innovative science and the protection of living Earth.